Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th students, and this is for 15th February 2021. As we are into the revision mode and we are revising the entire syllabus in the form of CBSE sample papers, and we have already discussed and reflected upon 3 CBSE question papers, and we are right now discussing question number 4 or question paper number 4, and in that already we have discussed section A and section B carrying 40 marks. In this session we are going to discuss about section C and part of section D. That is, section C, as all of you know, will have 5 questions and each question carries 3 marks, so it is worth 15 marks. So let's go straight into the question. 26th question number. If a true breeding homozygous pea plant with a green pod and axial flowers as dominant characters is crossed with a recessive homozygous pea plant with yellow pods and terminal flowers, then what would be the A. genotypes of two parents? So genotype of two parents, capital G, capital G, capital A, and small g, small a, both dominant and recessive. B. phenotypes and genotypes of F1 offspring. Plant with green pod and axial flowers, heterozygous, it becomes GG, capital G, capital A, small a. And then the phenotypic distribution ratio of F2. So since it is a normal one, dihybrid cross, it has to be 9:3:3:1. Of course, you can also put the checkerboard. Axial flower is to terminal flower is to axial flower is to terminal flower. Of course, with parts also you have to write. That becomes 9:3:3:1. So it is always better you explain the answer with the checkerboard or this. Okay, then it will be easy for you. So draw the F1, write the gametes. So the gametes from this is capital G, capital A, from this parent is small g, small a. So these gametes fuse and zygote formation will be capital G, small g, capital A, small a. This is F1, right? That's what we have said here. And then this has to be selfing will be done, and then you can put that checkerboard. Okay, 9:3:3:1, right? Question number 27. Now the sequencing of total genomes is getting less expensive day by day. Soon it may be affordable for a common man to get his genome sequenced. What could be the advantage and disadvantage of this development? Advantages of affordable genome sequencing. It can help in settling disputes which may arise in case of parentage of the child. Okay, biological parents will be detected. This can also help in disputes of property inheritance by finding bona fide beneficiary. The human genome can also help in preparing a database on people with criminal records. It can help in identifying the chances of genetic disorders in a family. So many advantages. But where there are advantages, there will be disadvantages also. What are those? Genome sequencing can have serious issues of privacy. Some employers may misuse the data to blackmail their employees. Many private matters may leak into public domain, creating embarrassment for the affected person. So privacy can be compromised. That is the thing. Question 28. For an organ transplant, it is an advantage to have an identical twins. Why? Organ transplant involves a critical issue of tissue rejection. This happens because the immune system always tries to reject any foreign substance. In case of identical twins, most of the tissues would be identical in both the individuals. Hence, chances of tissue rejection would be minimal if donor and acceptor pair is from identical twins, because genome is same, blood group also. Hence, an identical twin is considered to be an advantage for organ transplantation. Question 29. Describe the various evidences for genetic role of DNA. Evidences for the genetic role of DNA. It occurs in all cells. Thus, can act as genetic material. We have proved that through experiments also, that DNA is the genetic material. It is capable of replication, thus can form its own carbon copies. DNA copies are precisely the same as the original DNA, hence not changing the information. Fourth, the amount of DNA per cell is constant. DNA is much stable during cellular metabolism, which is necessary for genetic material. DNA regulates cellular activities through transcription and translation. And seventh, DNA is the main component of chromosomes, and chromosomes act as vehicles of hereditary information. Question number 30. What are the misconceptions about the transmission or acquiring AIDS? There are many wrong beliefs and misconceptions about the AIDS, acquired immunodeficiency syndrome. AIDS cannot be acquired by the following activities. 1. Insect bite, crowded transport, shaking hands, sharing towels, coughing and sneezing, kissing and embracing, sharing utilities and telephone, swimming pools and toilets. So these are the misconceptions that are there. Through these, there may be spread of AIDS. Next, there is a choice for this. Why is using tobacco in any form is injurious to health? Explain. Tobacco contains a large number of chemical substances, including nicotine, which stimulates adrenal gland to release adrenaline and noradrenaline into blood circulation, both of which raise blood pressure and increase heartbeat. Tobacco chewing is associated with increased risk of cancer. Of course, lung cancer is the primary one, and oral cavity, mouth cancer. Smoking reduces oxygen carrying capacity. So that is the main thing. And now we go to section D, which has got 3 questions. Each will be having 5 marks. So you have got 3 5 15 marks. So this is a long answer type, and I have told you whenever there is a long answer type, you better always select questions which are having like small sub questions like 1, 2, 3, 4, 5. Attempt that than a big descriptive type. Like 31, 1. How is oogenesis markedly different from spermatogenesis with respect to growth till puberty in humans? Oogenesis is markedly different from spermatogenesis in the following. Of course, you know spermatogenesis, it occurs in males starting from puberty till the completion of life cycle. Whereas in oogenesis, it starts before birth during embryonic development. Now look at that, before birth during embryonic development, and goes on until the lady reaches menopause. Next, second one. A single spermatogonium after second meiotic division, meiosis 1, meiosis 2, forms 4 haploid. Haploid means n number, one set of chromosomes. Spermatids that mature to form spermatozoa. So there is a diploid, 2 sets of chromosomes, then that becomes haploid. A single oogonium after second meiotic division produces only 1 worm, thankfully, and 2 non-functional polar bodies. So only 1 functional worm will be produced by a lady. The process of spermatogenesis, second meiotic division, completes in testes and releases matured sperms. That's spermiosis and spermiation. The second meiotic division of oogenesis completes in fallopian tube when sperm enters the secondary oocyte. So these are the notable differences between the spermatogenesis and oogenesis. Of course, second one, my favorite one, draw a sectional view of human ovary and label the different follicular stages, ovum and corpus luteum. This will be very helpful to you in practicals also. It is 3 mark question in practicals, and here it is almost 2.5 marks. So draw the beautiful diagram, keep on practicing diagrams as I have been telling you. So start with blood vessels, primary follicle, tertiary follicle. Of course, you can draw secondary follicle and tertiary follicle, and most important Graafian follicle, and breaking of Graafian follicle and release of ovum, ovulation, and then remaining, after the ovulation takes place, remaining layers becomes the corpus luteum, and you know the importance of corpus luteum. Okay, so keep on practicing diagrams, and particularly as I have been telling you, section C and section